Hello everyone, and in today's session we are going to talk about the prokaryotic cells. So let us begin with the topic. Yes, so millions of microbes are present around every corner, and certain microbes such as bacteria have nucleoid inside them. But such kind of bacteria do not have any membrane bound cell organelles. For example, bacteria, blue-green algae, mycoplasma, or PPLO, which is also known as pleuropneumonia-like organism. Also, one feature of prokaryotic is that they multiply rapidly as compared to the eukaryotes. Now, such kind of organism, we call them as the prokaryotic cells. So these prokaryotic cells that we are talking about, they vary greatly in their shapes and size. And they generally have four basic types. First one is the bacillus or rod shape, in which these bacteria are rod shape. Now the second one is coccus, in which bacteria are of spherical shape. Now third one is the vibrio, which means the bacteria are of comma shaped. And the last one is the spirillum, in which bacteria are of spiral shaped. So now let us understand about the features of prokaryotic cells. Now, all prokaryotic cell possesses a cell wall which is surrounded by the plasma membrane or the cell membrane, or also called as the semi-permeable membrane. But the cell wall is not present in case of mycoplasma. So apart from the cell wall, the fluid-like matrix which is present inside the bacterial cell is called as cytoplasm, in which all the contents of bacterial cells are suspended. Now apart from that, no well-defined nucleus is present, means the nucleus you see here, also called as nucleoid, it lies freely inside the cytoplasm. So here is another small circular structure present in the cytoplasm that is called as plasmids, and it gives the advantage to the bacteria, means it provides antibiotic resistance. Antibiotic resistance for this bacterial cell. Yes, so now no membrane bound organelles, as you can see in this bacterial cell, is present except for the ribosome, which you can also see lie freely suspended in the cytoplasm. Now, apart from ribosomes, various inclusion bodies, which are also called as mesosomes, and which are the enfoldings of the cell membrane, are also present inside the bacterial cell. So many microscopic studies reveal that most prokaryote, especially the bacterial cells, have chemically complex cell envelope, which are tightly bound, forming three-layered structure, consisting of first one calyx, second one cell wall, third one is the cell membrane. And all these membranes, all these structure or layer perform different functions, but together they act as a single protective unit for the prokaryotes. Bacteria can be classified into two groups. First one is the gram-positive bacteria, second one is the gram-negative bacteria. So bacteria that take up the gram stain in the staining procedure, that kind of bacteria is known as gram-positive bacteria, and bacteria that do not take up the gram stain in the staining procedure are known as gram-negative bacteria. Now, the gram-positive bacteria that takes up the stain, it turns violet, as you can see in this image, and the gram-negative bacteria that do not take up the gram stain, it turns pink in the staining procedure. So the reason the gram-negative bacteria do not take up the stain is because of the presence of lipopolysaccharide membrane present in it, which does not allow or which turns pink when stained with the saffron. So the presence of lipopolysaccharide membrane is the another reason that these gram-negative bacteria are resistant to the antibiotics. Means the antibiotics are not able to cross this membrane, and it provides better resistance in case of gram-negative bacteria. And that is the reason the gram-negative bacteria are harder to kill as compared to the gram-positive bacteria. So, the gram-positive bacteria, if we talk about, they take up the stain and turns violet because of the presence of peptidoglycan layer, which is very thick as compared to the gram-negative bacteria. Here you will find the single layer of peptidoglycan layer, and in the gram-positive bacteria, you will find the thick layer of peptidoglycan layer which takes up the stain. So, the example of gram-positive bacteria is Bacillus anthracis, and the example of gram-negative bacteria is Pseudomonas. So now let us discuss about the cell envelope. So cell envelope of bacteria act as a single protective unit but have distinct functions. Glycocalyx, for an instance, it resists antibiotics entering inside the bacteria. Also, it differs in the composition and thickness. For example, in some prokaryotes, the glycocalyx is thick, which is called as capsule, and in some of the prokaryote, it is present as a loose sheet, which is called as a slime layer. Now, glycocalyx is followed by the cell wall, which determines the shape and gives structural support to the bacterial cell, which prevents the bacterial cell from collapsing and bursting. Now, the cell wall is followed by the cell membrane, which is selectively permeable in nature, and it controls the entry and exit of various molecules. For example, food particles, chloride ions, sodium ions, and bicarbonates. So even though these layers have distinct feature, but together they act as a single protective unit. So often plasma membrane extends inside the cell and forms the mesozone, as you can see in this image. So this extension, as you see inside the cell, is called as mesozone. Now these extensions are seen in the form of either vesicles, tubules, or lamellae. Now the functions of these mesosomes are that they help in the cell wall formation, they help in DNA replication, also they help in the distribution of daughter cells. Apart from that, they help in respiration, secretion, and provide increased surface area of plasma membrane. But in case of cyanobacteria, another extension of membrane but containing photosynthetic pigment is also there, which is the character feature of this cyanobacteria and called as chromatophores. So the bacterial cells can be motile or non-motile. And if the bacterial cells are motile, they have extensions from their cell wall, which is called as flagellum, or in a plural form it is called as flagella. And this flagellum is composed of three parts. First one is the filament, because of which the bacterial cells gets the push to move forward. Second one is the hook, which is a flexible joint between this flagella and the cell surface. Now third one is the basal body, which provides the motion to the bacterial cell. Now among these three parts, the filament is the longest portion that extends from the cell surface of the bacteria. So many motile and non-motile bacteria have pili and fimbriae, which are the surface structure of the bacteria. But these fimbriae and pili do not play any role in the motility, means they do not play any role in the motion of the bacteria. Now, pili is the elongated structure, as you can see in this image, that it is the elongated structure which is made up of a special protein called as pillin. And fimbriae is a small bristle-like structure here arising from the surface of the bacterial cell, which helps this bacteria to attach to the rocks in the stream or the host tissues So inside of the bacteria is filled with cytoplasm that hosts the genomic DNA. And apart from that, cytoplasm also consists of ribosomes. These small blue colored structure that you see inside the cytoplasm are the ribosomes. Now, ribosomes measures 15 to 20 microns in size, and they are made up of two subunits, that is 50S and 30S, together forming the 70S prokaryotic ribosomes. Now, this is a very important point for you to remember that about the subunits of the prokaryotic ribosomes. Now here, the S you see stands for Swedberg's unit, that is for the sedimentation coefficient, and it is a measure of size and density of each ribosome. So these ribosomes are the site of protein synthesis. So these several ribosomes, basically what happens is the several ribosomes, those are attaching to a single mRNA. So here the red color long strand which you see is the mRNA, and the bigger structure, the pink colored structure you see here is the two subunits of ribosomes, and the clover-like leaf structure here you see coming and getting attached to the mRNA is the tRNA. It is called as tRNA. Now these three components together help in the formation of proteins from this strand of mRNA. So this whole process of conversion or formation of proteins from mRNA is called as translation. So the formation of proteins from mRNA in the presence of ribosomes is called as translation. Now cytoplasm also contains the inclusion bodies. That is, these are the reserve materials which are stored in the cytoplasm. And these inclusion bodies are not bound by any membrane system, but they lie free in the cytoplasm. For example, phosphate granules, synophysin granules, and glycogen granules. And other inclusion bodies such as gas vacuoles are found in blue-green algae, purple and green photosynthetic bacteria, which helps these organisms to float in the water. So now you see that prokaryotes are very simple organisms, yet they are able to thrive in any condition. So yes, with this we come to an end of our topic, that is prokaryotic cells. Hope you understand the topic well. Stay tuned for more amazing concepts. Thank you.